Assalamu alaikum, dear students. I am Muhammad Rauf. Welcome to the course Principle in Animal Life 2. This is lecture number 26. The topic is Transcription in Eukaryotes. Discussing agenda for the current topic will be Introduction, Central Dogma of Molecular Biology. Transcription is the DNA-directed synthesis of RNA, molecular components of transcription, synthesis of an RNA transcript. Lecture outcome. After watching and listening this lecture, students will know they will be able to describe the flow of genetic information from gene to protein. Introduction. The DNA inherited by an organism controls specific traits. Traits is any heritable feature like hair color, skin color, body color, height, and that particular information for the development of that structure or for the maintenance of that structure is present in the DNA. DNA directs the synthesis of proteins and of RNA molecule involved in protein synthesis. DNA carry instructions for the embryonic development of an organism, for controlling different traits in that organism, for the synthesis of proteins, for the synthesis of various enzymes used in biochemical reactions. So how this information is expressed? We know that enzymes perform function and enzymes are synthesized by the DNA. So what is the intermediate steps that results in the synthesis of enzymes? Proteins are the entities which perform different functions, and proteins are synthesized by the DNA. So how the proteins are encoded by the DNA? The information for the synthesis of a specific protein is encoded by DNA. So how DNA directs protein synthesis? That will be the focus of this lecture. Proteins are the link between genotype and phenotype. Genotype is the genetic makeup and phenotype is the physical outlook. So genes express physical traits through proteins, courtesy of proteins. Suppose a person has freckles, dots on his face. Now that information is there in the DNA to develop such a facial structure. But if you study the protein complement of that individual, then you will come to identify a specific protein which is responsible for freckles on the face. And this protein is actually the link between the genotype and phenotype. So gene expression is the process by which DNA directs the synthesis of proteins, or in some cases just RNA. Gene expression is that process in which the information encoded by a gene is expressed, and this information is usually expressed by the product of that gene. The product of that gene can either be a messenger RNA or other types of RNAs. Remember here that there are different types of RNAs like messenger RNA, transfer RNA, ribosomal RNA, siRNA, snRNA, and so on. But only messenger RNA is the only transcriptional product which is also translated. The other types of RNAs are not translated. They are transcribed and then after certain modification they becomes functional. Messenger RNA is the only transcriptional product which is also translated, and it is called messenger RNA because it carry message for the synthesis of a polypeptide chain. The expression of genes that code for proteins including two stages. Now this gene expression, when we talk about messenger RNA, then gene expression has two distinct stages, transcription and translation. Transcription is the synthesis of RNA using information in DNA, means the messenger RNA or other type of RNA is synthesized from DNA. The DNA is unwound at a particular site and then one of the strands act as a template for the synthesis of a messenger RNA through a process called transcription. So transcription produces messenger RNA. Messenger RNA carries a genetic message for the DNA to the protein synthesizing machinery of the cell. Once messenger RNA is synthesized, then after slight modification, if it is in a eukaryotic cell, it finds its way through nuclear pores into the cytoplasm where it forms association with ribosomes, and then with the help of transfer RNA and other enzymes, the information encoded in messenger RNA for the synthesis of a particular amino acid, particular polypeptide chain is decoded and polypeptide chain is formed. So this was the process of transcription. Now translation, remember that only messenger RNA is translated. The other types of RNAs are not translated because messenger RNA is the only RNA which carry message for the synthesis of a polypeptide chain. And ribosomes are the protein workbenches. They are the universal organelles. They are the structures on the surface of which the process of translation takes place. Here is an overview of the whole process. This is the process of transcription and translation in a eukaryotic cell. Why it's eukaryotic? Because the nucleus is covered by a membrane. So the process of transcription occurs inside the nucleus. Then the messenger RNA is processed. The 5' cape and 3' poly A tail is attached. Introns are removed and then it becomes mature and it finds its way to the cytoplasm through nuclear pores, and the information for the synthesis of a polypeptide chain is decoded in association with ribosomes and transfer RNA. Now, central dogma of molecular biology. This phrase was used for the first time by Francis Crick in 1956. He was the co-discoverer of the 3D structure of DNA, and he used that phrase, and it is called the central dogma of molecular biology, or the central dogma of life. And this phrase states that the transfer of genetic information in a biological system occurs through this path, where DNA encodes information for the development of an organism, or for the synthesis of proteins, and then a messenger RNA is copied, and from the messenger RNA a polypeptide chain is synthesized. So it is common to refer to gene products as proteins rather than polypeptides. Polypeptide is a strand of many amino acids, but proteins may be much more complex than a simple polypeptide. There are instances where the proteins are molded into tertiary and quaternary structures, where there is combination of several polypeptides. So it's better to use the word protein instead of polypeptide. In bacteria, translation of messenger RNA can begin before transcription has finished. Bacteria is a simple prokaryote. It lacks a well-defined nucleus, and furthermore, the life of bacterial messenger RNA is very, very short. So the process of transcription and translation occurs simultaneously at the same time, and furthermore, the bacterial messenger RNA does not require any processing because it is translated quickly and simultaneously, and the bacterial DNA lacks non-coding regions or introns. In eukaryotes, the nuclear envelope separates transcription from translation. Eukaryote RNA transcripts are modified through RNA processing to yield the finished messenger RNA. Means after transcription, the messenger RNA is called primary transcript. It undergoes modification, the addition of head and poly-A tail, the post-transcriptional modification in which the introns are removed. So after those modifications, the RNA becomes mature, and then it finds its way to the cytoplasm via nuclear pores. Now we will discuss the process of transcription in a bit detail. So messenger RNA is transcribed from template strand of a gene. An enzyme called an RNA polymerase separates the two strands of DNA apart and joins together RNA nucleotides complementary to DNA template strand, thus elongating the RNA polynucleotide. Now, this is a gene, a particular area to be transcribed. This is the promoter region of the gene. The RNA polymerase and the other transcription factors needs to attach to this position, and only then they will be allowed to synthesize a messenger RNA. So it's a very important step for the RNA polymerases and other enzymes, the general and specific transcription factors to attach to the gene at correct location. And once they are attached with the gene, then they can start synthesizing the messenger RNA, and then the end of the gene arrives and the process is terminated. So the process of transcription has those three stages. First, initiation, when the RNA polymerase and other enzymes come and attach to the promoter region of the gene. Second is elongation, where the actual messenger RNA is synthesized. The third is the termination, where the process of synthesis of messenger RNA is completed and RNA polymerase is released from the gene. Now here, once again, remember one thing, that a gene will always be transcribed from 3 to 5 region, means the orientation of the newly synthesized daughter strand will be 3 to 5. Here you can see this is a 3 dash, and this is a 5 dash, and now these are double stranded DNA. Now here the upper strand is being used for the synthesis of the messenger RNA. It's the lower strand that is being used for the synthesis of messenger RNA. And the strand which is used for the synthesis of messenger RNA is called template strand. But here one thing to remember is that RNA polymerase is different from DNA polymerase. DNA polymerase is responsible for the replication of DNA molecule. This enzyme is responsible for the synthesis of RNA from DNA, and the difference between RNA polymerase activity and DNA polymerase activity is that RNA polymerase does not require a primer to start polymerization, and it does not have the ability of proofreading. This is the disadvantage. So if it makes a mistake, if it inserts a wrong base while copying into messenger RNA, it cannot rectify that mistake. So during transcription, one of the two DNA strands called template strand provide a template for ordering the sequence of complementary nucleotides in an RNA transcript. The template strand is always the same strand for any given gene. We have already discussed that, but there are certain genes in bacteria where both the ends can be used for the synthesis of RNA, and such genes are called inverse genes. The RNA sequence where RNA polymerase attaches and initiates transcription is known as promoter. In bacteria, the sequence that signals the end of transcription is called terminator. Molecular biologists refer to the direction of transcription as downstream and the other direction as upstream, means the exact point from which the transcription begins. The nucleotides below that point, or the region below that point, is known as downstream, whereas the region above that point is known as upstream. And this is the point which is designated as 1, and the upstream bases are designated as -1, -2, -3. Downstream bases are designated with a plus sign. The stretch of DNA downstream from the promoter that is transcribed into an RNA molecule is called a transcription unit. Transcription unit is actually the coding area interspaced by non-coding regions of the gene. It is the actual area which has information for the synthesis of a polypeptide chain. Now, bacteria has only a single type of RNA polymerase, whereas eukaryotes have 3 different types of RNA polymerases, and of them, RNA polymerase 2 is responsible for the transcription of messenger RNA. The other RNA polymerases transcribe RNA molecules that are not translated into proteins. So here once again the 3 stages of transcription: initiation, when the RNA polymerase and other transcription factors bind to the promoter region; elongation, when the actual coding area is being copied to messenger RNA; and termination, where the process comes to its end. The DNA sequence where RNA polymerase attach is called promoter. RNA polymerase binding and initiation of transcription. Promoter signals the transcriptional start point and usually extend several dozen nucleotide pairs upstream of the start point. Promoter is a region which is located several nucleotides upstream of the actual region which is being copied. So here is the promoter sequence, and the promoter sequence has a Tata box. Tata box means there is thiamine, adenine bases. Similarly, there is another specific promoter sequence called CART box. So Tata box is located 25 nucleotides upstream from the transcription start site. Means if the transcription begins here, then Tata box is present here. 25 bases upstream of the start point, and this will be used as a template strand for copying. Several transcription factors, one recognizing the Tata box, must bind to the DNA before RNA polymerase II can bind in the correct position and orientation. So first, enzymes come and they recognize the promoter sequence, then they recruit the RNA polymerase and other enzymes to the promoter region, and once the initiation complex is formed, then the process of transcription can start. The completed assembly of transcription factors and RNA polymerase II bound to a promoter is called transcription initiation complex. A promoter DNA sequence called a data box is crucial in forming the initiation complex. In eukaryotes, a collection of proteins called transcription factors mediate the binding of RNA polymerase and the initiation of transcription. Only after transcription factors are attached to the promoter does RNA polymerase II bind to it. Here is a diagram taken from another book. This is the promoter region. Promoter region is the region where the initiation complex is formed. Enzymes, RNA polymerase, and transcription factors first come, and they first recognize this point. They attach with this point, and after attachment, when the pre-initiation complex is developed, then this whole transcriptional machinery starts migration in the downstream direction towards the actual area which will be copied. So then they migrate towards the transcription start point, and then they start forming the messenger RNA. The messenger RNA synthesis will take place in 5 to 3 direction, always in 5 to 3 direction. As RNA polymerase moves along the DNA, it untwists the double helix, exposing about 10 to 20 DNA nucleotides at a time for pairing with RNA nucleotides. The enzyme adds nucleotides to the 3' end of the growing RNA molecule as it continues along the double helix. In the wake of this advancing wave of RNA synthesis, the new RNA molecule peels away from its DNA template, and the DNA double strand reforms. So when RNA polymerase reaches the end of the gene and the coding area is all copied to messenger RNA, then the messenger RNA is peeled away, just like you peel the skin of a banana. In the same way, the messenger RNA is dissociated from DNA, and the two strands of the DNA are reannealed. And a single gene can be transcribed simultaneously by several molecules of RNA polymerase, following each other like trucks in a convoy, where several trucks are going on in a line on a road. In the same way, depending upon the need of the cell, if the cell is in greater need of the proteins or the transcriptional product of a particular gene, then The RNA polymerases are going one after another like a convoy so that more proteins are synthesized and more messenger RNAs are produced. The mechanism of termination are different in bacteria and eukaryotes. In bacteria, the polymerase stop transcription at the end of the terminator and messenger RNA can be translated without further modification. The transcribed terminator function is the terminator signal causing the polymerase to detach from DNA and release the transcript, which requires no further modification before translation because it's a prokaryotic mRNA, so its translation starts even before the termination of its transcription. Whereas the eukaryotic messenger RNA undergo modification. The modification can be the addition of the poly-A tail by the enzyme poly-A polymerase and the addition of 7-methyl GTP header. So this is the polyadenylation sequence. It is actually in the DNA and it is identified by RNA polymerase, and the RNA polymerase 2 transcribes the polyadenylation signal sequence. The RNA transcript is released 10 to 35 nucleotides past this polyadenylation sequence. So once the polyadenylation sequence is encountered by RNA polymerase 2, it means that the process of transcription will come to its end anywhere between 10 to 35 nucleotides ahead. This is an illustration from a book. The RNA polymerase is moving in the forward direction until it encounters a specific sequence polyadenylation signal where the process of transcription will terminate. And in polyadenylation signal, there are many adenine nucleotides which are copied to messenger RNA as uracil. So there is a long strand of 35 to 40 nucleotides consisting of adenine to uracil bonding, adenine on DNA to uracil on RNA, and because of this weak association, the RNA is detached from the DNA. And once RNA is detached, the double-stranded helical form is again reinforced. It was the end of the lecture. Thank you very much. Allah Hafiz.